So thank you for that, and I really want to give a shout out to Martin and his collaborators, not only just for the invitation to present some of our recent work, but for organizing this whole webinar series at a time when we all of our other meetings where we normally get together and see each other are on hold for a variety of reasons, mostly related to the pandemic. I think this has just been, although I haven't been able to get to all of them, I think it's just been invaluable for helping to keep our community together. So I'm really happy to be a part of it. And Martin and your folks, good job. Thank you very much. Okay, as many of you know, and as Alexei just alluded to, for many years we've been interested in the IP3 receptor calcium release channel, and along the way we became interested. If everybody could just mute their microphones, please. I'm picking up a little bit of a reverb. Mostly using electrophysiological approaches to study the single channel behavior of IP3 receptors, and along the way became very interested in the transfer of calcium out of the ER through the IP3 receptor into mitochondria, and how that regulated bioenergetics and some cell biology downstream of that. And so here is the issue. We now have some structural information about the proteins that mediate calcium egress out of the endoplasmic reticulum through the IP3 receptor, and calcium uptake through the mitochondrial uniporter, that calcium uniporter that I'm going to talk about today primarily. And what has been a focus of many discussions about this calcium transfer from the ER to the mitochondria is the spatial proximity. And so I've tried to show here in terms that reflect the spatial dimensions what this might look like, with the IP3 receptor in the ER on the left, and MCU and the mitochondria on the right. And one notion about this, and I'll discuss some of this, spatial proximity focus has been the notion that the intrinsic calcium sensitivity of the uniporter is very low, and so the mitochondria have to be localized close to sources of calcium, to be in nanodomains where calcium concentration is quite high. Here are some of the parameters that determine the magnitude of the endoplasmic reticulum to mitochondrial calcium transfer. We're talking about two ion channels, the IP3 receptor and the MCU ion channel. So the kinetic features, the channel gating, and there are a number of features associated with that. The thermodynamic driving forces and including buffering and the geometry of those contact sites. The calcium flux under physiological conditions, and then, as I just discussed, the spatial relationships among the channels. What we've been focusing on mostly is regulation of the kinetic features of the channel gating. What I'm going to focus on, and most recently by, well, over a long time actually, by calcium in the nanodomain, mostly on the IP3 receptor. What I want to talk about today is how the mitochondrial calcium uniporter is regulated by calcium in the nanodomain and in the matrix, and that'll be the focus. And what I want to suggest is that the uniporter is indeed regulated by calcium in the cytoplasm in a nanodomain, and that regulation is coupled to regulation by calcium on the other side of the inner mitochondrial membrane in the matrix. So here's a little cartoon depiction. I'll show some more recent structural information about the channel. So MCU is the pore-forming subunit. It's a tetramer with the ion conducting pore down the middle of that tetramer. In higher eukaryotes, its activity is 0 if this single pass transmembrane protein called EMRI is not co-expressed. EMRI then serves as a factor to promote the open probability. The MCU channel And it also couples through a physical interaction two paralogs, MICU1 and MICU2. In fact, MICU1 was the first protein discovered of this ion channel complex, and it associates with the complex in part through an interaction with an acidic C-terminal tail of emery. It forms an obligate heterodimer with MICU2, or in some tissues MICU3, but in most tissues it's the MICU1 heterodimer. In the absence of MICU2 or MICU3, MICU1 can also form a homodimer, and so depending upon the tissue and cell type, there could be MICU1 homodimers or MICU1, I think predominantly MICU1, MICU2 heterodimers. They each contain two functional calcium-binding EF hands, so a total in the dimer of 4 calcium-binding EF hands. And then finally, another protein that Magnesium discovered, MCUR1, is probably not a core component of the complex, but has something to do with regulating the channel. I'm not going to have much to say about that. And calcium then goes through MCU, driven by the very large membrane potential across the inner membrane that's generated by oxidative phosphorylation. Okay, so I want to just focus a little bit on some of the regulation of the transport through MCU by MICU1. So here's a little assay that I'm going to show from our lab. This has been done by other labs, in which I think these were HEK cells. We put them into a cuvette where we can measure the extracellular calcium concentration, and we start with an extracellular calcium concentration that's really quite low, close to basal cytoplasmic calcium concentrations, and permeabilize the cells with digitonin, and then add thapsigargin to take the endoplasmic reticulum out of the picture for any kinds of changes in calcium that we'll see. And under these conditions, and I'll show some evidence for that in just a second, the changes in calcium in the bath are due to mitochondrial transport mechanisms. So at this point, we've reached a steady state after we let this transient release of calcium from the ER relax. We're down at pretty low calcium concentrations, not so high compared to basal resting levels. And at this point, mitochondria are in a steady state where any calcium influx through the uniporter is balanced by calcium extrusion through the sodium-calcium exchanger. So at this point, we can add this compound, the CGP compound, to disrupt the pump leak balance by eliminating the activity of the sodium-calcium exchanger. And this enables us to then get an instantaneous picture of what's going on with calcium influx. And if you do this in cells that lack MCU, you see that nothing happens, because mitochondria can't take up calcium because MCU is absent. You'll also notice that in wild-type cells, MCU seems to be not active. On the other hand, if we do the same experiment in cells expressing MCU but lacking MICU1, you can see that the uniporter is constitutively active under these conditions, in which in the wild-type cells expressing MICU1, or in cells in which that have been rescued by re-expressing MICU1, but not in cells that were rescued by expressing MICU2 only. So this is absolutely dependent upon MICU1. There is a constitutive leak, constitutive activity of MCU in the very low calcium concentration regime. And so we discovered this with Madesh Muniswamy around the same time that Chodash and Hajnashki did it. The data I'm showing is some recent data that Riley Payne in my lab did. We called this MICU1 inhibition of calcium uptake in the low calcium regime. We called it gatekeeping. And gatekeeping means that the open probability, with no real mechanistic insight, that the open probability of MCU is very low. And in fact, we're carrying these out for a really long time. I would say it's it's very low, if not zero. Another way you can do this experiment is shown here. So here we've basically done the same type of permeabilized cell experiment. Now we're adding little boluses of calcium at around 700 seconds into the experiment. And you can see in the black traces in the wild type cells, they really don't take up calcium until you get over a micromolar, and then even over a micromolar, it's still not very robust. On the other hand, if MICU1 is not part of the complex, the cells take up, the mitochondria take up calcium robustly. And so MICU1 mediates inhibition of the channel in the low calcium regime. Sorry, I'm having trouble keeping. Don't know how we got to this. It's my daughter. Okay, sorry. All right. So this, when MICU1 is not part of the complex, the MCU is constitutively active in the low calcium regime, and you can measure mitochondrial calcium concentration, and we've done that. We've using a genetically encoded indicator. You can see that basal calcium in the mitochondria is constitutively elevated. In fact, there are patients that have loss of function mutations that demonstrate the same phenotype, and there is muscle and brain disorders related presumably to this phenotype. And this has been observed in a few different patient populations now. So this was a kind of a simple-minded model of for the regulation of MCU activity by cytoplasmic calcium concentration. So in the low, under resting calcium concentrations, I'm depicting MICU1 and MICU2 as one complex here with 4 calcium binding sites, the EF hands, coupled to the channel through its interaction with emery as well as with MCU itself. And the little plug on the left indicating no calcium flux when calcium in the cytoplasm is low is not to represent any type of mechanism. This isn't to suggest that MICU1, MICU2 are blocking the pore or any mechanism. It's to suggest that the open probability of the MCU channel is low. And I'll come back to this a little bit later. And then when calcium rises, there's a conformational change shown here in this little, depicted as this little triangular piece in the MICU1, MICU2 heterodimer that enables the, that is sensed by MCU, and in the MCU open probability is enhanced, allowing calcium influx. I keep losing my, so sorry. I'm not sure what happens here. Okay, so we have some structural information about this. So this is from Fan et al. Just published this year. This is what it really looks like. This is a picture of the holo complex. There were pictures of various fungal complexes, and then eukaryotic complexes without MICU1 and MICU2, but here's what it looks like. And so the stoichiometry is 4 MCUs form the pore. The red dot is a calcium ion sitting in the selectivity filter. 4 emeries are associated with each monomer. The monomers are each associated with 1 MICU1, MICU2 heterodimer. So the stoichiometry is 1 MICU1, MICU2 heterodimer to 4 MCU to 4 emery, i.e. one channel has 1 MICU1, MICU2 dimer. And very interestingly, and this has been observed in a few different labs now, these monomers can exist as either monomers structurally or dimers, with both having functional activity. The dimers associating through their N-terminal domains. So whereas there's a lot of symmetry in the transmembrane domains, which would be up here, this would be in the matrix, the N-terminal domains are distributed asymmetrically into a clamshell-like shape, and then in the dimeric structure, they interact, and if you disrupt the interaction, that breaks the dimeric structure. So this is a strong, the N-terminal domains are a strong determinant of the dimer. Another determinant of the dimer, although you can break this interaction without breaking the dimer, is MICU2. So MICU2 has two interfaces. One interface is, oh geez, where'd it go? Am I presenting? I'm presenting. Where am I? Sorry. Still see this? Yeah, okay. MICU2 has a MICU2 interface, as well as an interface with MICU1. MICU1 interacts with MICU2, and it also interacts with the channel, mostly through interactions in the presence of calcium, mostly through interactions with the C-terminal tail of Emery, shown here, with some minor interactions with MCU. So this structure was solved in the presence of calcium when one expects that the channel should be opening, permitting calcium flux. This is just another structure that was determined earlier by Wang et al., in which MICU1 and MICU2 were not in the complex. And I wanted to show this just that you could, because they suggest that this could be localized at cristae junctions with the outer membrane, positioning it in very close proximity to the cytoplasm of cells. So here's my depiction of what I just showed you. Here is the complex. The MICU2s are shown in red, MICU1s are in green. You can see that MICU1 is associating with, there are really only two, there are 4 Emerys associated with the channel, but only 2 are really participating in the interaction with the MICU1, MICU2 heterodimer when the channel is open. And what I also show is the calcium ions sitting in the selectivity filter immediately at residue E264, immediately underneath another set of residues, D261. These will become important in just a second, and I highlight them here to show that MICU1 is certainly, when the channel is open, not associating with this this is a calcium binding site. The real selectivity filter is immediately below it at position 264. So here's, if we twist that structure around, you can see that the channel is just wide open to the intermembrane space. You can see the calcium ions sitting down in the selectivity filter right below this row of aspartic acid residues at 261, with MICU1 and MICU2 sitting way off to the side of the channel, with 2 Emerys shown here in orange and cyan, coupling the heterodimer to the channel. A very different structure is observed in the absence of calcium. Here now, the entire MICU1 heterodimer has rotated around and sat itself down on top of the channel so that it completely occludes access of the channel pore to calcium in the intermembrane space. And so here's my own little depiction about this. This on the right is what I just showed you. Here are the 261s, calcium sitting below it in the selectivity filter. And in the absence of calcium, when we know that the channel is inhibited by the MICU1, MICU2 complex from the functional studies, we can see that the MICU1 is associated very strongly with the acidic 261 residues, occluding the pore, so the calcium permeation through the channel is impossible, forming a structural basis for the functional studies that I just showed you. Okay, so this does look like it's kind of a pore block mechanism, is the gatekeeping mechanism that I talked about. But I think this model is too simplistic. And I'm going to go through a lot of different, I'm going to go through a lot of reasons for why I think that is. First, it doesn't account for cooperative activation of the channel. Hajnowski's lab was the first one to show this. This is some data that Riley Payne in my lab showed. You can see that in a wild type cell, activation by cytoplasmic calcium concentration is strongly cooperative, and the total activation, the total flux through the channel, or the open probability of the channels, is higher than is able to be achieved by simply knocking out MICU1 and MICU2. So the channel, there's more to channel activation than simply removing, in my opinion, than simply removing the block by MICU1 and MICU2. That's one problem. The second is work that has come from our lab by Horia Vai, in which we've used patch clamp electrophysiology to study calcium currents more directly. So here we've used the protocol first developed by Yuri Kirchuk and David Clapham's lab, in which we can go into the whole mitoplast mode after we break the outer membrane, dialyze the matrix with whatever we want. In this case, importantly, a very highly defined calcium concentration with defined buffering capacity, and then we can change the calcium concentration in the bath in order to measure calcium currents going through the channel. And so here's just an example of that. We ramp the voltage from -160 inside the matrix to +80, and you can see inwardly rectifying currents that are dependent on the concentration of calcium in the extracellular medium and are all ruthenium red inhibited. So these currents are not observed in the absence of MCU in these cells. These are HEK cells. So these are MCU calcium currents. And so here's a second reason now based on electrophysiology data, and this was really first described by Yuri Kirchuk, and I'll just show some of Horia's data here. The channel can open with MICU1, MICU2 unliganded. So if you take calcium away from the extracellular side, it becomes sodium permeable. You can measure linear sodium currents. In this case, MICU1 and MICU2 is unliganded, and yet the channel is open. So the model of an apo MICU1, MICU2 forming a simple channel block cannot account for these data. The second The second aspect that model doesn't account for is the fact that matrix calcium concentration also regulates MCU activity. So here we've either put with 1.5 millimolar EGTA as the buffer, either no free calcium concentration or 100 micromolar, and you can see that the currents are comparable in those two situations. But at an intermediate concentration of calcium, at around 400 nanomolar, you can see that the calcium currents are strongly inhibited. And in fact, there's a biphasic, as these data suggest, relationship between the calcium concentration in the matrix and the amount of current, or the open probability of the channel, with an apparent KD for inhibition of around 60 nanomolar, a peak inhibition with 1.5 millimolar EGTA at around 400 nanomolar, which isn't all that much different than the free calcium concentration in resting mitochondria, which is about 100 nanomolar or so, and then a recovery from that activation, which is highly cooperative. So this suggests a new model in which we postulate that on the matrix side, there's an inhibitory site and an activating site. And so here's kind of a little schema for the way the channel might work when the MICU1, MICU2 EFNs are liganded by calcium. When cytoplasmic calcium goes up, the channel opens because of a conformational change in MICU1, MICU2. Calcium fluxes through the channel. Calcium then can bind to an inhibitory calcium binding site. And despite the fact that MICU1 and MICU2 is calcium liganded, the channel closes. And that's not accounted for in any model right now. And we would suggest that implies that there is a conformational change in MCU emery that enables the calcium-bound MICU1, MICU2 to interact with the channel. And then when calcium concentration gets somewhat higher, the lower affinity activating site can be liganded, and then there must be a different conformational change, or it relaxes back to the same conformation in which the calcium liganded MICU1, MICU2 is no longer able to inhibit the channel. So matrix calcium controls the functionality of MICU1, MICU2. So where are the matrix calcium regulatory sites? So in many plasma membrane calcium ion channels are regulated in the nanodomain created by calcium flux through the channel itself. And one way to distinguish whether, where the calcium is to use a very fast high affinity buffer like BAPTA and compare the responses to a slower calcium buffer like EGTA. So everything that we've done so far has been in EGTA, and so we switched to BAPTA to ask, does the calcium that's regulating the channel actually come from the calcium that's permeating through the channel itself? So the prediction was that if we introduced BAPTA, that we should shift this biphasic curve to even higher calcium concentrations, or maybe even eliminate the whole calcium matrix calcium regulation of the channel completely. That was the prediction, that this is what BAPTA would do. What we found instead, so here now we're using the same concentration of BAPTA as we did for EGTA, and you can see that instead of being inhibited at 400 nanomolar, now it's actually inhibited at lower calcium concentrations in the matrix, exactly opposite to what we predicted. So here's what we see with 1.5 millimolar EGTA, and here's what BAPTA does. As we increase the concentration of BAPTA, instead of shifting everything to the right, it shifts the biphasic relationship stays intact, and it shifts everything to the left. So it sensitizes, increasing buffering capacity sensitizes MCU to matrix calcium concentration. And in a log plot, that's a linear, semi-log plot, that's a linear relationship suggesting that there's one dominant process that is being regulated by BAPTA. And so we had to come up with a more complicated model. We call this a flux sensor because, by analogy with plasma membrane calcium channels, as I suggested before, maybe this BAPTA is telling us something about a site that's responding to calcium coming through the channel, and that this sensor is then regulating these two other sites that are coupled. And we suggest they're coupled because this biphasic inhibition and escape from inhibition remain intact over the entire range of calciums and buffer concentrations, and that this flux sensor regulates the apparent affinities of those two sites. So in the presence of calcium, when those sites in the presence of EGTA, when those sites become liganded, the apparent affinity of those sites goes down, and so the biphasic curve is shifted to the right. On the other hand, when buffering is very strong and that site remains empty, the apparent affinity of those sites gets very, very high, and the sensitivity of those sites shifts way to very low calcium concentrations. That model explained, can account for the data except for this data point here in the presence of very high concentrations of BAPTA. What we expected was, because we have to record with high calcium concentrations in the pipette in order to record sufficient current densities, we expected the channel to be, the MCU to be, MICU1, MICU2 to be fully liganded, all of these sites to be unliganded. We expected the channel to be open, and yet it was inhibited. It was behaving as if the inhibitory site was liganded. So it occurred to us that even though we were buffering all of the calcium, that we had magnesium in the pipette solution, and that we hypothesized that magnesium may be interacting with the site. And we remembered a study from Peter Stathopoulos, who described in the N-terminal domain a magnesium binding site, and importantly coordinated by, I'll point out a couple of residues that I'll talk about, 131 and 147. And so we wanted to test whether this might play a role in the inhibition of the currents that we saw when we had 5 millimolar BAPTA, no calcium, but we had magnesium. You can see as soon as we take the magnesium out, then there's no inhibition of the channel, suggesting that maybe this site is playing a role. And so here we've taken MICU1 knockout cells, rescue them with MCU knockout cells, rescue them with MCU, added MICU1 because Riley Payne has determined that in order to get stoichiometry right, we have to do it this way. You can't just overexpress MCU. You can see that we can recapitulate in these studies the inhibition by 400 nanomolar calcium, but if we mutate that one of the key residues in MCU, express a mutant MCU, that inhibition by matrix calcium is abolished. And that's just shown here. Similarly, if we mutate the 147 site, inhibition by matrix calcium is abolished. So this suggests that site plays a role in the matrix calcium regulation that we're observing. We asked, all right, we're seeing this in electrophysiology. Does it happen in intact mitochondria? So we use the same type of protocol that I described before in permeabilized cells. And in the green are wild-type cells. We're measuring calcium in the bath and mitochondrial membrane potential simultaneously. You can see when we add a pulse of calcium, about 5 micromolar pulse of calcium, mitochondria take it up very, very quickly because MICU1 inhibition is relieved. There's a transient depolarization associated with that. And we can, I only show 5 pulses here, but in these cells, we can add 9 to 10 to 12 pulses before mitochondrial calcium overload and activation of the permeability transition. On the other hand, when we mutate, when we do the same experiment in cells expressing either one of these N-terminal mutants, we can only add 2 to 3 pulses of calcium before they undergo what appears to be permeability, calcium-induced, overload-induced permeability transition. So we think that this is an important feedback mechanism in intact mitochondria. Finally, I alluded to the fact that it looked like matrix calcium regulation was controlling, how MICU1, MICU2 were able to was able to regulate the channel. So we looked at that a little bit more explicitly by disrupting the interaction of MICU1, MICU2 with the channel, either by mutating the C-terminus of Emery. So here, deleting all of, neutralizing all of its charges, or knocking out MICU1 or MICU2. And you can see that this matrix calcium regulation, conversely, depends completely on MICU1 and MICU2 on the other side of the membrane. That can be, as shown in the bottom, completely rescued by re-expressing the wild-type versions of any of these proteins. And so here, that's just shown again. Here's a different mutant of Emery where we just completely lopped off the C-terminal tail. You can see this normal inhibition that we see at 400 nanomolar calcium is completely abolished if MICU1 and MICU2 are not part of the complex. So in conclusion, MICU1 and MICU2 play, I haven't really shown this, but distinct roles in gatekeeping and cooperative activation. And this provides a mechanism for cytoplasmic regulation of the uniporter. But matrix calcium also regulates MCU activity, mediated by what appears to be at least 2, 3, maybe calcium binding sites in the matrix. And this regulation is coupled to the calcium regulation that's mediated by MICU1 and MICU2 on the other side of the membrane. And this type of regulation then provides a finer feedback, control of MCU activity, and suggests a very important role for matrix buffering capacity in regulating the activity of the uniporter in different cytoplasmic calcium regimes. And with there, I'll stop just to give a real big shout out to Horia Vai, who did all of the electrophysiology that I just showed. So thank you very much. I'm sorry that ran a little bit long.
Okay. Thank you very much, Ken. So I'm now looking at the questions. So maybe I can start. Can you hear me, Kevin?
I can, yes, thank you.
Very good. So I think that there is a kind of range of publications indicating that mitochondrial calcium uniporter is not the only mechanisms of calcium entry into the matrix. So there is a kind of number of the publications which are actually kind of different cell types, different methods of measurements. We actually have also seen it in our laboratory. The influx is tiny, but it exists. And I just wonder whether you have seen electrophysiological correlate of this alternative mechanism.
Jorge Vai could speak to that most clearly. The way we define the MCU-mediated calcium current is by its inhibition by ruthenium red, relatively low concentrations of ruthenium red. There still is a background current, but we haven't explored, and it's very small by comparison with the MCU current in the cells that we've looked at, but we haven't explored whether what that current is. In the permeabilized cell measurements we do, it's very striking to me. I mean, it has been suggested that maybe that technique isn't sensitive enough to see alternative calcium influx pathways, but you can look for a very, very long time, and that calcium in the bath, in the absence of sodium calcium exchange activity, really doesn't change. So we've only really looked at one cell type. So I really can't comment on other calcium influx pathways.
Great, thank you very much. There is a question from Martin. So, are the mechanisms described conserved in all cell types?
Yeah. So, I mean, one of the some of the pushback I've gotten from this has been from a lot of people have if we're talking about the matrix calcium regulation, I don't know. We've been just focusing on, although we like to get out of this cell type because of the genetic models that were afforded to us in this 293 cell type, we've only really looked at this matrix regulation there. So one idea we have right now is to look in other tissues, other cell types, mitochondria and other cell types. There is a notion, this is from Liron Boyman and John Lederer, that even in cardiac myocytes, for example, that even the MICU1, MICU2 kind of gatekeeping mechanism doesn't exist. There are other strategies to limit calcium influx. For example, they suggest that a low density of MCU channels is all you need, as long as you have a functional sodium calcium exchanger. Many labs have reported the MICU1, MICU2 regulation of mitochondrial calcium influx, and there is a cardiac phenotype in mice lacking MICU2. So I don't know. We do want to look at this whole question in cardiac myocyte mitochondria, but I don't think, there are some pieces in the literature. I think Anat Parekh has contributed to this. Klaus Vollheim has contributed to some of this literature. Maybe you have, that suggests that using very, very different approaches, that there is matrix calcium mediated feedback inhibition of calcium uptake, which we recognize to be primarily through MCU. So I think, yes, we haven't shown it, to this kind of extent in other mitochondria.
Okay, thank you. So the next question is from Mohammed Trebak. So he's asking how magnesium enters the matrix.
Yeah, well, I think Madesh Munaswamy just had a publication about this, but it's been known for a while that there's a magnesium selective ion channel. I think it's MRS1, MSR1, MRS1. It's magnesium selective. And I think that's the primary mechanism for magnesium uptake in the mitochondria. Somebody else? Yeah.
Mm-hmm. So Donald is asking about the possible distinct roles of MICU1 and MICU2. Do they have independent roles?
Yeah, so Riley Payne published a paper on this where we really tried to parse out what each one was doing, and Kim Kamer in Vamsi Mutha's lab has done very important work along these lines. In fact, she was often one step ahead of us. But when we've looked at the behavior of gatekeeping, both in terms of the threshold that it sets and the cooperative activation, there's a very distinct difference between MICU1 homodimers and MICU1, MICU2 heterodimers. And we suggested that what MICU2 does is tune both the gain and the threshold. So the threshold actually gets higher with MICU2 in the system, and the gain is shifted to lower calcium concentrations. So they're playing very, very different roles. And the structural, the structures now provide they don't address that specifically, but they do suggest that MICU1 is the dominant mechanism, and that's why MICU1 homodimers can work, because MICU1, but not MICU2, can associate with the intermembrane space aspect of the channel. And what MICU2 is doing is then presumably, because it interacts differently with MICU1 than MICU1 does, through some type of allosteric regulation that's not informed by the structures so far, because there is no structure with a MICU1 homodimer in it, alter that interaction. As I said, the structures still don't provide evidence for how the channel is cooperatively activated. They suggest a simple, pop the lid off of the can kind of regulation, but there's got to be more than that, and MICU2 impinges on that.
Okay, thank you. So Natasha Privarska asked you to comment on the role of MICU1, MICU2, and MCU in mitotic catastrophe.
Yeah. Mitotic catastrophe is, as I understand it, cells go into mitosis and then they kind of explode. As they're kind of going through mitosis. I don't know I don't know what's happening to calcium flux through.
I have no idea.
Through MCU under those conditions. Perhaps the question could be rephrased to kind of jog my memory about how I should be thinking about this.
Okay, so next question is from Antonio Villalobos. Tumor cell mitochondria have a great capacity to accumulate calcium without swelling. Could the regulatory mechanism that you have described be different in normal and tumor cell mitochondria?
Yes, what I've suggested actually in this final slide, we've changed buffering capacity and kinetics with EGTA, with these artificial buffers. In the paper that we published in PNAS about this, we also showed results of some very difficult experiments in which we tried to use the normal buffer phosphate. I think, exactly what the calcium concentration, we haven't measured calcium concentrations during these experiments that I showed you when we were talking about matrix calcium regulation. Physiologically, so for example, when we knocked out the important calcium binding sites in the N-terminal domain in the 131 and the 147 mutants, we weren't measuring matrix calcium concentration, so I really don't know how high it goes. Under resting conditions, matrix calcium concentration is about 100 nanomolar, same as it is in the cytoplasm. When we're adding these pulses of calcium, the rate of calcium uptake is once the membrane potential has gone back to where it was before we added the first pulse, is very comparable in the second pulse, suggesting that after we've already loaded up the mitochondria with some calcium, it's not really affecting the driving force, which suggests that it's no longer free, that it must be bound. And so if there are, in different cell types, different buffering capacities, I think that would play a very important role in the mechanisms that we're talking about here. Yes.
I think that Mohammed Trebak is asking about your your interpretation, your thought about mitochondrial calcium unipolar being able to buffer calcium entry through crack channels. How can mitochondria possibly fit into the tight Erpm junction?
Yeah, well, I think we've been interested in the IP3 receptor and the ER to mito communication. Anything that we could have been talking about to the extent that MICU1 and MICU2 is expressed and playing a role in the majority of mitochondria in cells, then you have to satisfy the calcium liganding requirements of MICU1, MICU2 before the channel can open. So, and since we've defined that and others have defined that as being in the 1 to 2, 1.5 micromolar range, you have to be pretty close to the source of calcium, because most agonist-induced changes are kind of are less than that. So, yeah, depending upon how close the mitochondria are, then, I mean, if they can't fit into that space, then they're not in that nanodomain. So I can't really say much more about it than that. And, yeah, I mean, except to say that if a mitochondrion is close enough to a source of calcium, then everything we're talking about applies, I think.
I think that Jonathan Sobolov is commenting that mitochondria don't fit into Erpm junctions, but they could sit right outside. I think that actually triple junctions were described. So mitochondria, plasma membrane, and ER junction.
Good.
So I think that Geert is asking, do you envisage effect of calcium on MCU complex via cardiolipin? And he refers to Madesh work.
I'm sorry, the question is, do I think cardiolipin may play a role in channel regulation?
In calcium dependent effect on MCU.
On the matrix calcium dependence?
Well, I think that in this particular case, the question is calcium, but I presume that this is matrix calcium.
Yeah, there was a structure from another group. I'm blanking on who it was right now. It was a Chinese group that solved the MCU structure in human MCU structure in higher resolution and showed the heavy investiture of the channel by phospholipids, but importantly by many cardiolipin molecules. And there is functional studies from Madesh with Vamsi-Mutha. I don't know what, I can't recall the other functional studies, but it looks like these lipids are intimately associated with the channel, fitting into crevices, for example, between TM1 and TM2. So we haven't played around with that. You know, in the electrophysiology studies, this is a native membrane, so one assumes that the cardiolipin that is normally associated with the channel is still there. In fact, everything we've done has still been intact mitochondria, but we haven't tried depleting cardiolipin or taking advantage of those structures to ask how could we disrupt the interaction between cardiolipin and the channel. So I don't know the answer to that.
I think that Mohammed is commenting that close, kind of, if they just close, they can't sense calcium, basically stating that close is not good enough, but maybe we can discuss it later during the kind of free discussion. There is a question from Li Zhang. So she asks, do other ions affect MCU activity?
Well, we would suggest from the matrix side that magnesium is can interact with that inhibition site that we would suggest is normally. I'm a little yeah. So definitely. So try to keep this short and simple. Magnesium definitely on the matrix side. On the cytoplasmic side, I think those EF hands are so far relatively selective for calcium, but somebody can jump here and correct me if I'm wrong about that.
So,
the inhibitory, what's very interesting about this matrix regulation through the N-terminal domains is the fact that if you look at the structures, all of those N-terminal domains are different. They have the same structures, but because they're not symmetric, two of them are sitting on the outside of the clamshell and two of them are sitting on the inside of the clamshell. And so it occurs to me that it may be possible for calcium and magnesium to simultaneously be interacting with different N-terminal domains in a single channel because those N-terminal domains are not created equal. I should also point out that there was another structural paper recently of N-terminal domains in the dictyostelium MCU channel that had a completely different structure, which oligomerized into a tetramer spontaneously, with the tetramer being completely dissociated by calcium, suggesting that what we would suggest then is that calcium binding through the N-terminal domain, somehow that information is propagated up through MCU or Emery to the part of the channel that's interacting with MICU1, MICU2. That conformational change in the structure where MICU1 and MICU2 are interacting with MCU in the presence or absence of calcium binding to MICU1 and MICU2, so in those very different structures with the lid on or the lid off, don't show any change in the MCU, in MCU itself. And so I think there's more structural work to be done if our thinking about this is correct, to observe these structures that enable, for example, in this conclusions in this slide here, for a calcium liganded MICU1, MICU2 to interact with the channel.
Okay, excellent. So maybe if I can't see more questions, maybe I can ask a very brief question. So I think that ATP-ADP ratio can change magnesium concentration. Do you think that this could be kind of physiological, at least one of the physiological mechanisms of responses to hypoxia, something like that?
You mean matrix calcium? Matrix,
this is matrix magnesium.
Matrix magnesium, yeah. Matrix magnesium?
Yeah, so if you basically change ATP-ADP ratio, magnesium concentration will change. Of course, you need to change it substantially, but
we haven't. Yeah, so we haven't added. We don't have any ATP or ADP in our patch clamp experiments. So it's free magnesium, and obviously inside the matrix, magnesium would be complexed. A lot of it would be complexed with ADP and ATP. So good point. Yeah, we should try that experiment.
Yeah, so whether this is whether the changes of magnesium during acute hypoxia are sufficient, and whether this is the magnesium channel or another sources of generation of magnesium which could drive this mechanism.
Yeah, good point. Yeah, I don't know.
Okay, I think that there are no more questions, and time is appropriate to say thank you very much, and maybe back to Martin to activate the discussion.